Yes, Mr. Siddharth.
Yes, sir.
Please introduce about, sir. Yeah.
Thank you so much, Professor Rajmohan Balakrishnan, sir. Now, the next session delivered by Professor Anjani K. Tiwari, Associate Professor, Baba Saheb Bhimrao Ambedkar University, Lucknow. He is also a ex-scientist E of Institute of Nuclear Medicine and Allied Science Defense Research Development Organization, Delhi. Dr. Tiwari is lifetime member of ICNM, SNMI, ISN, ISCA. He is also a many more achievements and fellowship. Without taking more time, I would like to invite Professor Anjani K. Tiwari for his blessing and kind word about the topic role of PET MRI for understanding of neuron glial interaction.
So please, sir.
Yes, sir.
Thank you, Dr. Siddharth. I think I'm audible to all.
Yes, sir. Of course, sir.
And slides are sharing, I think.
Yeah, it is sharing. Yes, sir.
So first, I would like to thank the organizer and our friends and colleague, Dr. Himanshu, who has invited me to give this presentation, a little interaction for the role of PET, which is positron emission tomography, and MRI, that is magnetic resonance imaging. These are two basically biomedical techniques which is normally used for detection of different disease state as well as normal cellular stage condition to different modalities. Now, before I start, I would like to share some of the glimpse of my university where I am working. So once the pandemic conditions over, I would request or I would like to invite all of you in my university, which is located near to the airport, Lucknow airport, around 4 to 5 kilometers. So after pandemic, I would like to invite all of you to my university whenever you all get times. Okay, now I start that if I'm saying for neuroglial interaction, it means we want to see how the brain cells are working. I mean, how they are interacting. So if you remember our 12th class lectures, we know that there are some type of cells which are normally known as microglia, astrocytes, dendrites, oligodendrites. So basically these interacts, and then we are able to do all the functions which is responsible for our daily activity. Now the question is how much we are able to understand the biological function of our body by these modalities. So first, before that, if you remember one more point, that what was the radiation range which we have studied in our physics class. So the range started from cosmic rays, X-rays, ultraviolet, infrared, and radio rays. And energy-wise, the maximum was for cosmic rays, and then it was decreasing. So if we are saying that X-ray modality, definitely it is going to use X-ray. If we are saying for PET or MRI, so PET will use gamma and X-ray region, and MRI will use radio region. So definitely the radiation burden of positron emission tomography and single photon emission tomography is much than the MRI. It means if a person is going for some scan through CT, X-ray CT or PET or SPECT, he will take more radiation burden or he will take more radiation in his body during a scan. But in comparison to this, if you are going for MRI, definitely the radiation burden will be less. So normally that is the reason why doctors say, okay, you should go maximum 2 or 3 scan in a year, because more than that may harmful for your body and cells. Now, with the start, what is biomedical imaging? If I will recall, or you all can recall, कि एक बचपन में हमें कहा जाता था कि what is image or what is shadow. So once the light is coming to some object and it is not able to pass that particular object, you can get the shadow of that material. If we are able to construct that shadow in 3D image, I mean along with X, Y and Z axis, it can construct an image. So this was the basic for image construction. So initially it was for 2D signals, like in we are using 20-30 years back for X-ray. And if you are using 3D or 4D images, it is the computational tomography which we are using. So it's a non-invasive visualization of internal organs. We want to see our the internal organs functioning and their surroundings through non-invasive techniques. That is the role of biomedical imaging. Now, what are the medical technologies? They can show your tissue at organ level or at cellular level. Even at molecular level, you can also detect those things, like you can calculate how much density a particular receptor is there. Even you can calculate that how much neurotransmitter are crossing through one space. I mean, the latest research is going in this direction that you can calculate at a particular time that, okay, how much neurotransmitter are working at a particular space or a particular sub-region of the brain. So if I will talk the starting, the concept of this was started in 1895 and through X-ray, and it was also awarded Nobel Prize in 1901. And just read the last line. Within 1 year, I mean, after the invention of X-ray, you can get 1,000 publication of Röntgen rays, which was called at that time. So see, what was the research aspect of that X-ray? Nowadays, if you will click on Google or PubMed, you will get 20,000 publication per year or 20,000 different work in this field, at least for these biomedical modalities, for life science application or for human applications. Now, as I said, that initial X-ray will give only two-dimensional. And if we go for MRI, so MRI is just an extension, or we can say 3D extension. MRI is just 3D extension which we have studied in NMR. Normally in NMR, we look for proton or the hydrogen. Now it is MRI is 3D extension of those proton. Now someone may ask, to which proton you are asking. So in our body, there are different biofluids. And if I'm saying biofluid, definitely they are also made up of H2O. So those proton which are rotating our body in the form of biofluid or other fluid, their signals we can get. So if a cell is working normally, the pattern of biofluid will be different. And if a diseased cell is there, or if a cell is growing in a diseased state, definitely the pattern of that biofluid will be different. So accordingly, we will get different images in MRI, magnetic resonance imaging, based on the computational tomography or 3D projection. In this, we are also taking 3D projection of those protons. Now, how it is important for understanding the brain function. So one of the aspects is functional MRI. Like we have heard about yoga. We have heard about, the brain activity. So the functional MRI normally uses oxygen versus non-oxygen regions in the brain, and you can get the information like, okay, these, if you are performing some particular task, and while performing, what are the changes of the level of oxygen in different subregion of the brain, you can detect that. In terms of level of oxygen, this is one aspect which we can detect by functional MRI. Similarly, bold is another technique, and diffusion MRI is another technique to understand what information by this one. So one of the technique I have not mentioned in this slides, MRS technique, if we are going for a spectroscopic technique. So if there is disease state like Alzheimer's or Parkinson's, so definitely these are neurodegenerative states. So if neurodegenerative conditions are occurring, some of the biofluid or some of the chemical constituent which is found in our brain, that will change at that time. And if you are detecting those things, I mean, if you are able to detect those chemical constituents through spectroscopy, which is known as MRS, then you can identify a pattern for all the disease: primary stage, middle stage, and final stage. So these are the basics of MRI. So MRI basically gives information in terms of oxygen level. If you will use fMRI or the bold concept. So another one is diffusion MRI, and third one basically we normally use MRS, spectroscopy. What are the chemical constituent which is changing during neurodegenerative or other diseased state, or even in some trauma or some brain injury. So this is the aspect of MRI. And the second thing which I would like to say, another one is more promising: positron emission tomography. In terms of quantification at molecular level, this technique is much better than MRI. But in terms of radiation burden, definitely in this technique, the radiation burden is high. If a person is going for a scan, usko radiation burden zyada milega. So definitely there should be balance. If you want to quantify the receptors or any chemical constituent in our body, PET has more capability. But in terms of radiation, MRI has more capability. Now, I believe that there are a few students which are from graduation or PG level. So I will discuss some of the basic aspect of PET. So PET is also one of the non-invasive diagnostic imaging technique which can measure the metabolic activity of cell in the human body. As the name itself explains, that positron emission tomography. It means a tomographic techniques which rely or which based on positron emission. Now, simply someone may ask that how positron will emit. Unlike MRI, MRI uses the proton of the biofluid in general condition, though you can use other chemicals also like lanthanide, like gadolinium complexes, but for that purpose biofluid is also enough. But here you have to take some radioactive substance which is able to generate positron, because the name is positron emission tomography. So what is the basic, if I will say what is the basic of that technique. So if you remember one of the basic concept of class 9th, that in the periodic table there are 112 elements. At present you can say 120 or 124 elements, and around 2000 isotopes are available which we can detect or can see in general, though the others are lesser stable. So if we plot a graph between N by P, neutron upon proton, we will get three regions. Some of the substance where N by P means neutron upon proton ratio is 1, they are called as belt of stability, elements of the belt of stability zone. Some where N by P ratio is more than 1.5, they are unstable like actinides, जो कि pumps and इसमें use होता है. And the third category where N by P ratio is less than 1. So in these three regions, the stability is in the middle zone, belt of stability, which is known as belt of stability. So 1.5 also want to come in this zone to regain more stability. Similarly, less than N by N upon P ratio, they also want to come in this region. So what are the ways for those isotopes in which N by P ratio is less than 1? Either they can capture K electron or they can emit positron. Example. For example,
98 था कितना था?
For example, 611. So here the neutron upon proton ratio is 5 upon 6. So this is able to generate positron, and in that case, once it emits positron, the ratio will become more than 1. So this is the way of generation of positron. So in MRI, we are using biofluid or the protons of the biofluid molecules. Here we are using those isotopes in which N by P ratio is less than 1. Now what will happen? Once a substance or once a radioactive substance which is supposed to be injected as a PET marker goes in the body and able to release positron, it means it will release positrons and electron of that biofluid will mix with it. So positron and electron are antiparticles and they will combine and they will produce two photons at 180 degree. I mean positron and electron will combine and they will produce two photons at 180 degree because we have to conserve energy also. So if you are able to detect these two photons, then we can get the image of PET also. So this is the principle of PET, that we can detect those photons at 180 degree and can construct 3D images. So what are the things which we have achieved in this one? The first molecule which can detect, that is FDG, fluorodeoxyglucose. Most of our body cells, or we can say 99.99% cells, are using glucose for metabolites. So if there is some discrepancy in their function, either lower metabolism or higher metabolism, we can get the information of those metabolism. Similarly, about all the receptors, there are some selective ligands which will give information about those conditions. One of the example is, Parkinson's and Alzheimer's disease, where dopamine plays most important role. So you can easily detect the concentration as well as the density of dopamine transporter as well as receptors. Either it's a D1, D2, D3, similarly for serotonin. So if we are able to know the receptors, that okay, these are the receptors or these are the neurons which are responsible for this one, we can get the information, and accordingly we can plan for the therapeutic aspect also. So MRI gives the information about the oxygen level of a particular cells in the brain, as well as the chemical constant which are changing in the brain and their diffusion pattern. And the PET gives the information about their metabolic, I mean those cells which are interacting are in disease state. The metabolic aspect, glucose metabolic aspect, their receptor density, as well as how the neurotransmitters and transporters are working. So based on these, we can give the hypothesis, or we can analyze our hypothesis, okay, like how the glial cells and astrocyte cells are interacting based on their thermodynamics as well as their quantification and density pattern. Now, the one question, okay, if these two modalities, if I mean PET and MRI are so important, are so, I mean, fascinating, are we able, or can we try to merge them? Now, if we will try to merge them, which was started in 2015 onwards, there are few problems. Like PET detectors uses gamma ray, MR radio. So MR has lesser energy. Definitely MRI will use magnetic coils, and PET detectors will collect the gamma rays. So the energy difference is there. So we need few materials which are compatible in those two regions. So there are few questions, I mean, to merge these. Similarly, if a function, I mean, if a modality is working in radio region and another modality is working in gamma ray or X-ray region, the software requirement should be different, because accordingly the collimators and other system has to arrange or adjust into each other. So these two requirements are the limitations. That's why the PET MR system, I mean combined system, because everybody is saying, okay, it's a multi modal era, means one thing should combine in the second modality and second modality should combine in third modality. They can give more information. Like if you're going for CT one time, another time you are going for MRI. So if the modalities are merged, the scan, the number of scan will be less. So these are the latest attempts by the researcher and scientists to combine these modalities, that we can get all the information at one platform. I mean metabolites, chemical constituent, receptor density, transporters, and neurotransmitters stage. So if you are able to get all information at one platform, definitely we can understand more about our brain function as well as how they are working in complex and normal scenario. Yeah, these are the few of the challenges which I was discussing, and these are some of the initial prototype which has been prepared, and now some of the machines are installed in our country also as well as the other countries, but still it is limited for peripheral organs. In brain still there are few limitations which we hope that it will be solved in near future. Thank you.
Okay, sir. Hello.
Hello.
Yes, sir. Thank you very much, sir, for the outstanding illuminative talk. It is quite more relevant to the focal theme, because we are, as mentioned, the recent trends in life sciences, and it is the interdisciplinary aspects with the chemistry and its intervention with the life sciences. So it's filled with very and all the students present in the seminar, I suppose, have learned a lot, because they are most learned persons at the early stage. just pass out from BSc because they have good concept of the chemistry also. Therefore, they can utilize such kind of techniques in the biological sciences, in the molecular biology, along with the microbial aspects for its greater exploration of the life science for the human beings and its welfare. Now I would like to invite Professor and Head Department of Biotechnology, Professor Sharma sir. Please invite the speaker.
Thank you, Professor Tiwari, for such a lucid presentation and letting us aware about various intricacies in the magnetic resonance imaging and its role, profound role in diagnosis. So it's indeed a pleasure to congratulate you, and also this lifetime fellow award from the International Association of Zoologists. Many congratulations from all of us for this particular felicitation. And thank you very much for agreeing to deliver such a lucid talk at this platform. Thank you, Dr. Anjani.
Thank you. Thank you very much.
Thank you so much, sir.
Thank you.
Now we have with us the most enthusiastic personality, young personality, has worked from different institutions in India and across the boundary. Dr. Manoj Singh, eminent nanobiotechnologist. He has recently received award